So first, thank you for three really interesting talks. I think one commonality of all of them that sort of was Bob's question, and I'll take another panelist to do the same thing. All of you sort of had diagrams of the process, and we could spend days sort of digging down. I'd like to turn to David's, and I think, clearly a lot of people want to know the value of patient input. And I sometimes am a little bit turned off by asking for that because it's so obvious. But I think, your contribution to that will be useful. But I think what we really do need to do is take that whole drug development process that you showed, that Citi developed, each of those steps and say, now, where is the most value and how can we achieve it? And I'd like to suggest that we start with some low-hanging fruit that costs very little because value is relative to the cost. And I love that slide. I actually took a picture, but I don't think it came out, of all the different great things Pfizer's doing. And they've put a lot of staff in place. All those things cost a lot of money. But what I would recommend as a baby step that could have a lot of value at a low cost is actually involving experienced patient advocates in your external stakeholder meetings. So, I like to distinguish between what I call the ladies' auxiliary model of patient involvement and what I think of more substantive patient involvement. You can spend a lot of money putting on these new layers and having a ladies' auxiliary of patients that you talk to about what you're doing. I don't think that I think the cost-benefit proposition there is pretty low. But if you take someone who's been involved in, let's say, the Cancer Clinical Trial Consortia or other sort of pretty sophisticated patients, and you engage them with your other external stakeholders, you start getting conversations where different stakeholders understand the trade-offs and really participate in a way that I think you'll have much more value. And I think the industry has been pretty late on that. Whereas in other funded trials, like government trials, they're further along. And specifically, then, if you look at the whole development, the informed consent or something, I really did like your innovations where you got people trying different things. In a previous life, I did human factors work for Bell Labs. That's sort of a no-brainer. And you already do this with doctors. You put together kits for, tissue collection and stuff. So, of course, you want to do that. But engaging patients, I think about the specifics where you're going to get the value. So I know that was more of a soapbox than a question, but thank you.
No, I think the comments are great, and so just to. We're actually, we don't have huge numbers of resources internally doing it. It is still sort of a nascent within many organizations that this is sort of a necessary thing, and we're always sort of, finding, money in the sofa cushions to do a lot of the things that we do. But that aside, I took, the whole concept of dare to try, the idea of, doing some targeted experiments. Evidence is evidence, right? And so it's whether it's evidence in a clinical trial or evidence in support of the things you're trying to do in a clinical trial. And that's, I think, the only way that you can convince your organizations or other people that these things actually do have value. So I love the idea. So we sort of tried that with the NPV work, which was to take a couple of use cases in there, but we have to actually go after these other things and then, put them in the real world and then say, here's actually what we saw in a real world example. So
but you don't really look for the value of bringing clinicians to your external stakeholder meetings. You don't have to do those equations. So I sort of resent that you have to do it for patients, but I understand. And the reason I say that the industry, not necessarily Pfizer, but Pfizer for sure, are throwing a lot of money at it is because I see lots and lots of people appearing as speakers who have in their title now something of patient engagement at a drug company, which I am, of course, an endorser of. but I think use that money wisely.
Yeah, I mean, we have to justify our expenditure at every step of the way, and so it's not that as a matter of fact, we do weigh whether or not we bring we bring other experts in for for other types of meetings. But it is but you're right. I accept your point that. We ought to be putting more and more resources to it because there is the real value is there.
Jeff.
Yeah, Jeff Ginsburg from Duke. So I think this question is maybe predominantly for Robert, but others certainly chime in. So drug exposure is a dynamic process, as is diseases and biology. Yet today we've talked about DNA, which is inherently static. But I haven't heard much about the other omics, the role of the transcriptome, proteome, metabolome, in some of the vision that you have for augmenting the drug development process. At the very least, that gives you endophenotypes. Like Sex paper was really about endophenotypes. It wasn't really about disease so much. And at very most, it could give you, exquisite insights into biology. So just as an aside, we recently discovered a completely novel mechanism of action for aspirin, and its antiplatelet effect that is independent of COX-1 by measuring aspirin exposure, doing aspirin exposure studies and measuring the platelet transcriptome in healthy volunteers. So why aren't we doing more of that type of work in early stage clinical development than at least I've heard about today or heard about anywhere for that fact?
Yeah, so maybe I'll kind of break it down into three parts. How you can use these sort of multi-omic approaches to find new targets. Second is how we can actually use those same approaches to get some sort of pharmacodynamic biomarker. And then the third is around getting those subsets of patients that may help us identify those populations for which drugs are more likely to work. I think in this space, this is where oncology has been incredibly successful. And I think the work that's happening in immuno-oncology is just totally fascinating. So, of course, I work for Merck. We have an immuno-oncology drug, so I want to make sure that these biases actually are set up front. But to actually find gene expression signatures, patterns of mutational burden, and other simple things, just like is something expressed on the surface of cells or not, PD-L1, as an example. I mean, you start to see these very orthogonal patterns of actually predicting who will and will not actually respond to the drug. Then marching backwards, you can then use those to actually try to understand early pharmacodynamic reads. Will a drug work or not? And then finally, you can actually walk back again to the very beginning to say, well, maybe we're identifying those patient populations that are resistant to treatment with anti-PD-1 therapy, and that can then unlock new drug targets. So all of that is actually happening, certainly in oncology, and it's incredibly interesting. I think maybe to your question of why don't we see it more, why aren't we seeing it in the real world, part of it just has to do with the complexity of diagnostic platforms in the real world. I mean, if you had a fancy RNA expression signature, how many community hospital settings can actually do that? It's actually very problematic. And then suddenly you're now faced with all these sort of commercial considerations. Do we want to have our drug approved in that patient population, et cetera, et cetera?
Well, I'm actually more talking about insights into biology that which was one of your major points is that we're not ready yet to really advance this paradigm until we have more biological understanding.
Yeah, so I think I think it is incredibly useful for understanding. I think we have to differentiate between cause and consequence, and I think many omics profiling approach, and I would say the microbiome fits into this category. You're not really sure if it's a cause or a consequence of disease. It clearly changes with disease. It's clearly correlated with disease, but is it a cause or a consequence? And so I think when I think about any mechanism to get at any profiling approach to get at fundamental biology, I always try to think about causal pathways and what can differentiate cause from consequence. Gene expression profiling, in a patient with rheumatoid arthritis, what's cause and what's a consequence? It's really hard to know. Germline risk factor that tells you that if you perturb, TYK2, IL-6, some of these other things, that occurred just by definition prior to onset of disease. So by definition is causal. We may not understand the mechanism, but it is a causal factor.
Actually, I had one more comment and question for Dr. Brooks, which is, one of the things that's been driving me crazy in 40 years of practicing genetics has been this nihilistic approach that it's okay to make a diagnosis for a disorder where we know what the underlying genetic causes are and not find out what the patient's genotype is and make that information available. And I think what you're stressing is that making the diagnosis is nowhere near enough. You need to know the genotype if you're going to do these sorts of basket trials, because it's across diseases. It's genotype specific, not necessarily gene specific or phenotype specific. You need to know the mutation if you're going to try to suppress it. So I'm wondering if you want to comment on whether our current system is geared up so that we can make the case that patients should actually demand to know their genotype when they have a genetic disorder, as opposed to saying, oh, okay, you know, quote, it won't make any difference, close quote, so we won't do it.
I completely agree and see the value in that, and I think often patients do get that information in some cases. They may not, in some cases, be fully aware of the implications of it. I think there's an issue of education there, and better access to one's information about one's medical condition, I think, is absolutely a good idea.
But there's a lot of resistance from third-party payers, for example, to pay for genetic testing when, quote, it won't make a difference, close quote.
Yeah, I think I see your point. I think that's an area that I'm not as familiar with, but in principle, I think there's tremendous potential value there. Like I said, probably more than people realize. And the more dissemination of the information, I think, in general, is a good idea.
Anyone else approaching the microphone? No? Okay, I have actually, Dr. Brooks, I have a question for you. So in your model for basket trials and bringing rare disease organizations in, when are you thinking of bringing them in? And I ask, so are they getting on at the point when a trial is being designed? What about natural history? And then I think our last speaker brings up the issue of quality. I was hearing from different patient groups where a gene would be identified, and then people would think, they wouldn't realize they didn't have the mutation, and so they were submitting things thinking they were eligible for a trial, and there was a lot of back work and assessment because it was a space that didn't have a registry. So are you thinking that the rare disease groups should come together and be part of some groundwork with registries before you do the SME trial model?
I think that in general would be a good idea, and we have some efforts ongoing to facilitate that within our office, and certainly our Rare Disease Clinical Research Network is an example of a network where we do have a lot of patients coming together for various reasons. I think in terms of, I don't think it's necessarily absolutely essential that happen first to get into one of these basket trials. It could be a situation where sort of, if you build it, they will come. If there was a trial ongoing, say, take for an example, a stop codon read-through drug that someone's developed, any individual patient group, and I'm thinking of the ones I work with, I mean, you could fairly easily go through each of those patients and see if they have stop codon. Someone could test their cells, and if they do, they could potentially get into that trial. And I think that's sort of the opportunity. I don't think that involves a great level of coordination across groups. I think that coordination across groups is a great idea for many reasons, but I think for this, it's probably not absolutely essential.
Yes, hi.
Yeah, hi. So Marnie Falk from Chop and Pen. I'm staring at the title of the talk, and I'm reminded that we're almost done, so I'm sorry to ask the last question. But I'm hearing a little bit of a, I don't know if it's ambivalence or a struggle. We want to use genetics in clinical drug development, but we want to use it to fit the common way you guys do things now. And I really appreciate the talk that said things need to change, but it's my impression, and again, I'm not in common complex diseases, that doing studies of 10,000 people in 12 continents, or what, not continents, but 12 countries, is still kind of the way pharma thinks. So, and rare disease research is still sort of the purview of academia and NIH. So I guess my question is, if we're really going to do it, don't we need to start thinking about each person? Like really think about, precision medicine in each person, kind of at almost like a different level, like the way we think about each rare disease patient? Because I understand diabetes is kind of lumped, right? And asthma is kind of lumped, and I understand that that's the historic way to do it. But I guess I'd be interested in your thoughts, because I'm sort of struggling to put it all together in my head of how is the drug industry going to change? And I work with a lot of small pharma, and I get it that they're changing, but the big industrial leaders, so all the guys that are here today and women, how is it really going to change? How is each person as a rare disease or as an individual really going to be brought in to change the process? Because right now all I'm hearing is trying to fit what our new technologies are into the current process.
So, I can, speaking for Pfizer, we have a rare disease unit that is very nimble, and so they're involved in Duchenne and Huntington's and other rare diseases, and I think they operate actually in a pretty nimble way. Organizational complexity, we build these apparatuses in these organizations to deal with, I think, the legacy of what we have been dealing with, right? So big cardiovascular studies and diabetes. And we have operations that are meant to sort of serve those kinds of studies. And we've had instances where, the ideas around small studies can get crushed under the infrastructure of what we've set ourselves up to do to try to meet regulation and all these other things. So we're adapting, and I think adaptation takes time. But you're absolutely right. I agree with everything that you've said, and I think we're improving, but we still do have these big, sort of the big boat taking a long time to turn, I think we're trying to do things like through Transcelerate, for example. I don't know if you're familiar with that. It's a cross-industry collaborative where we're trying to address these kinds of broad industry challenges that we've thought that we've had to solve as individual companies, but in fact that we can solve together because they're pre-competitive. They're not, it's not about compounds per se. It's about how do we make sure all of our investigators get trained and we can accept each other's training. And so you don't have to take training 12 times if you're conducting 12 studies for, different companies and those kinds of things. So we're trying to work together to get to, I think, a place where we're operationally able to do some of the things that you're talking about.
Thank you.
So under new models too, I wonder, David, what was your reaction as you heard PJ presenting this potentially new model? Would that make a space more attractive to a sponsor, where you've got this basket trial that's organized with potential infrastructure that you could either join, help develop, or plug into, and de-risk? Is that something that you see as a de-risking opportunity?
Yeah, I certainly do. I'm not responsible for portfolio level decisions within my company. So I don't, that's above my pay grade. But ultimately, I think where there are good opportunities, especially where we can learn and then apply that to other things that we're trying to do, I think those are the best kinds of investments that we can make.
A very brief question, Greg Fierro from JAMA, and this is a naive question. Are trials on clinicaltrials.gov at all indexed by variation or mutation? I was just looking at the search functions on clinicaltrials.gov and not seeing anything that vaguely looked like that. And it seems like a very simple thing to do that might facilitate some of the basket trial ideas.
I think that if there's some inclusion criteria that are required to actually enroll patients, then it would actually be part of the inclusion criteria. But I think unless you actually know that up front and pre-specify it, I don't think it's included. But you could take ivacaftor from Vertex, the cystic fibrosis drug. I mean, they were recruiting very specific patient populations that were genotype specific because that's how they developed the drug. So I think there are those examples in rare diseases and there are those examples in oncology. But I think the more open-minded, that's part of the challenge I think that the field faces. There aren't a lot of strong genetic predictors unless you develop the drug for that underlying mechanism.
Yeah. And the other thing about clinicaltrials.gov is that it's based almost completely on what's in the protocol. So unless it's stipulated in the protocol, then it's not going to be on clinicaltrials.gov.
We're going to start wrapping it up.
Yeah, we need to wrap the session up, but NLM is actually doing a project to understand what we need to make clinicaltrials.gov more useful, and so I think it's an opportune time to bring this to their attention. So with that, let's thank our speakers, and we'll move into our wrap-up session led by Laura.